Great. Yeah. Okay. Then I will start. Welcome. Good morning. This is Lydia from BMKGENE. I'm thrilled to welcome each one of you to our latest webinar. Today we will explore Amplicon in BMKCloud introduction. Our webinar today will last for about 30 minutes, followed by a dedicated 5 minutes for your questions and answers. You can freely submit your questions by using the Q&A button at the bottom of your screen. We are looking forward to receiving your inquiries, and if any questions are left unanswered, we will contact you to reply via email. Also, we'll share the slides and the recording of this webinar via email. In this webinar, we will explore the application of Amplicon sequencing on BMKCloud, a comprehensive bioinformatics platform. The following topics will be covered. Firstly, introduction to BMKCloud. Discover the powerful bioinformatics platform offering a range of tools, applications, and data mining services tailored for microbial research. Secondly, key results from Amplicon sequencing, a deep dive into the main findings and insights generated with BMKCloud's Amplicon analysis workflows. And thirdly, customizable data visualization and mining. Learn how to personalize visualization outputs and conduct your own in-depth data mining to meet specific research needs. This session will empower you to maximize the potential of BMKCloud for your microbial diversity projects, giving you hands-on knowledge to enhance your research efficiency and data analysis capabilities. Whether you are new to bioinformatics or looking to optimize your workflows, this webinar will provide valuable insights to advance your studies. But before we dive in, let me introduce our distinguished speaker for today's webinar, Johnson. Equipped with extensive expertise in bioinformatics analysis, shines in his role. Holding a master's degree from Beijing Forestry University, China, he embarked on his professional journey as a bioinformatics analyst post-graduation, specializing in high-throughput sequencing data analysis, especially Pacific biosciences data. Johnson honed his skills with precision and dedication. His career trajectory led him to BMKGENE, where he transitioned to the Overseas Bioinformatics Department. Here, he assumed the pivotal role of Cloud Platform Product Manager, steering the team towards innovation and efficiency. With a blend of technical prowess and strategic vision, Johnson continues to make significant contributions to the field, driving advancements in bioinformatics with unwavering determination. So let me hand over this virtual stage to our Master Johnson.
Thank you, Lydia. Hi, everyone. It's Johnson. Today, we will talk about Amplicon application in BMKCloud. It will contain introduction, results, and customization. I will first introduce to you about BMKCloud and demonstrate how to use BMKCloud step by step. First of all, you should know that BMKCloud can offer a one-stop solution. We have bioinformatics tools such as image generator, sequence analysis tools, and data processor. You can use online analysis applications to do some basic data analysis. BMKCloud provides various analysis applications. After you got your final report, data mining or customize image without application you can do. You can change colors, reset groups, or other customized settings by yourself. So, how does BMKCloud provide service? Let's start from data. There are three situations. Situations number one, sequencing and analysis completed in-house. Number two, only sequencing conducted in-house without bioinformatics analysis. And number three, for users with their own data. For data sequencing in-house, we can push data or report into your BMKCloud account, and you can try function online by yourself. For the users with data on their own, you can upload your data into BMKCloud by yourself. But we recommend opting for data analysis on BMKCloud to avoid significant time consumption of data transmission and instability caused by the network fluctuation. Let's recognize BMKCloud first. This is the screenshot of the main interface of BMKCloud. You will see the same page after you log in into our platform. Using navigation bar on the top, you can manage your account, change language, connect us by email. But actually, you can connect our cell manager. Next part, you can see your account main information, including your storage space left or analysis limitation. Navigation bar on the left is very powerful. You can find almost every function of BMKCloud here. You can manage your project, such as upload or delete data, monitor your analysis task here. You can also find analysis applications of BMKCloud in bioinformatics. And I will introduce this to you in detail later. Let's leave this aside for this moment. Today's presentation mainly includes the following three parts. Number one, task management. We will talk about task submission, task monitor, and how to download final report. And the second part is the main results. We will go through about the Amplicon application results in BMKCloud. And third part about customize. We will do some advanced analysis and using the image tools step by step. This part, I will tell you about task management. If someone wants to complete an analysis, we should prepare data, do analysis, and download results. In BMKCloud, specifically sequencing in-house, you can simply divide the whole pipeline into three parts, including task submission, task monitor, and download report. Now, I will demonstrate to you about Amplicon analysis using BMKCloud step by step. ensuring that you can finish the Amplicon analysis by your own. Let's turn to BMKCloud homepage. Here you can see is the main interface of BMKCloud. I introduced this page some minutes before. And on the left, you can see here, bioinformatics. Bioinformatics apps, click here. All the analysis application you can use will listed here. Today, we will use application as an example. Click this and open the analysis application. Here is the main page of task submission. There are many parameters you can change. On the top, you can change the project name. For your analysis further, we can use a project name such as 16 and today's data. Create this project and you can find all the results in this project. There the basic information, you can change the report name and the analysis version, pipeline version. Raw data updating will input the data already on the BMKCloud. If you have the data by your own and the data is not already on the BMKCloud, you can upload the data you should upload the data into BMKCloud first. We have data already on BMKCloud, so I can click here. You can choose the folder containing the data of Amplicon. Here I will choose data. Our BMKCloud will automatically compare the pair entries, such as here I have the 3, 4, and 2 paired 1 and 7, 4, 1, 2 paired 2, and they will automatically paired into the one sample. Other parameters such as the sample margin and Amplicon type is based on your analysis, on your experimental procedure. And you can change this in your situation. But what if someone don't have the specific data and want to try our BMKCloud Amplicon? What should they do? We prepared an example data or you can call this demo data for Amplicon. You can just click here. Demo data will not allow you to change the parameters, but you can get the final report and using the advanced analysis tools. So if you don't have some analysis data prepared, you can just use the demo data and give it a shot. After you change all these parameters, you can submit the analysis. And here, click the submit here. The Amplicon analysis will be submitted. The first step where we will talk the finished. We talked about how to submit an analysis. The second part, I want to talk to you about how to monitor this project we just submit. Let's back to the homepage of BMKCloud. Here, you can see the navigation project. I have many products here, so this will take a little bit long. This page will list all your analysis product here. Of this product, we have today's data containing is the product we just submit, and the label here, project tasks. Click here, and you can see all the running pipelines will listed here. This part have the label, meaning shown as the complete. If you have the analysis just to submit, this label will into a red yellow background and it contains running. If you have this running label, which means your analysis is still running and not finished. If the label here is complete, your analysis is complete and you can get your final report. You can monitor your analysis in this part. And after you got the final report, you can just check the final report and download. I have a pre-prepared analysis report here. Just click it. A new window will be opened and all the results will listed here. This is an HTML format report. This is an HTML format report is listed here. And on the left is the navigation bar of advanced analysis. We will talk about this part later. On the top, there are navigation bar of your parameters and download. You can download the final report in three formats, just as HTML format, all results, and PDF format. HTML format and PDF format contain the same things in different format. Is this page in HTML or in the PDF format? But this true format contains no middle statement results or the middle files, such as the FASTA file or the statement table. If you want all the analysis results and the middle files here, you can click all results and you will download a zip file containing all the results and the file, including inside your analysis. So we talked about how to submit, how to monitor, and how to download the result. Let's back to my slide. Let's summarize here. We talked about submit analysis using BMKCloud. We choose Amplicon applications. You can choose applications you need. Select the data in BMKCloud and set analysis parameters. All the applications in BMKCloud are using in this way. And we talk about how to monitor tasks, how to tell whether process is running or finished, and manage all your tasks. After finish analysis task, you can then download the final report into your personal computer. You can choose any format you need, HTML format, PDF format, or total zip file. Now this part, let's go through about main result of Amplicon application of BMKCloud. Here, the main workflow. Here is the main workflow. Let's just talk about bioinformatics part. We have data process, include data filter, quality control, merge, and assessment. For main results you may be interested in, BMKCloud provide OTU analysis, species annotation, alpha diversity, and bit diversity results. For some analysis design, such as with including environmental trees or group settings, BMKCloud provides some advanced analysis results. Data process and OTU opt-in. Raw data was processed as described above, and data quality was estimated based on raise lines, counts of raise at each target. OTU refers to cluster of sequence used to define a group in studies or population genetic studies. we can apply it to cluster reads with simply above 90-70%, generating OTUs. Text annotation of feature sequencing was processed by Bayes classifier using Silver as reference database. We can get statement of reads count in each text level, and statement of species annotation by this approach. And species annotation. After we got specific annotation results, we can do further visualization results and evolution results, such as a heat map of species richness cluster, evolution tree, or specific distribution bar plot. These images help you present your results better. Alpha diversity reflects the richness and diversity of species within a single sample. It can be assessed by several metrics, including many methods. BMKCloud provides all these results, and you could choose one suits your analysis designs better. In alpha diversity for each method, BMKCloud provides not only statement metrics in total, but also results in detail. You can check images and table for more information. For in bit diversity analysis was processed by software to compare species diversity between different samples. There are 4 common used statement methods to calculate distance between samples in bit diversity analysis. And here's the results of PCA and UPGMA. After that, we can combine multiple results and get results such as sample heat map analysis and UPGMA clustering tree and history combining diagram. For differential analysis between groups, if you have group citing, BMKCloud can provide your differential analysis results between groups, such as analysis of variations, diagram, or function gene prediction. You can try this function by yourself. Notice that statement analysis methods are complex and diverse. They are with different principles and applications. Please select the analysis method and the results that can best explain the biological phenomena according to your actual needs. Let's summarize here. We went through whole results quickly, and you can see the BMKCloud provide most of your needs. I just showed a little part of whole results, and I suggest that you can try BMKCloud on yourself. Using demo data or yourself data, check the report. The demo data analysis is free. After we got the final report of Amplicon, you may meet these situations, such as I don't like the color site in the report, or I want to change the group settings, or I want to analyze alpha diversity by myself. Then you can do this using advanced analysis tools in BMKCloud. You can find function in BMKCloud divided into 2 parts, functional module and analysis module. For functional module, you can view your report, monitor your advanced analysis, or update report, such as remove samples, change group settings, or change analysis parameters. And for analysis module, you can do advanced analysis, may be part of the main workflow. We can use alpha diversity as an example. Select this function, and you can change analysis parameters as you wish. BMKCloud can automatically use data with analysis, which analysis is needed. Then you don't need to choose or upload table or matrix into the analysis platform. Then you can submit analysis, monitor, and download the results. Now, I will demonstrate to you an advanced analysis using BMKCloud step by step. Let's turn to BMKCloud. Here is the main interface of analysis report we just obtained. On the left is the navigation bar of the advanced analysis tools. I just introduced to you about we have the functional module and analysis module. First part I want to show you is how to update the report. If you don't act, if you set the parameters wrong or you want to remove some samples, you can use this function in the updated report and parameter settings. Here, you can change all these parameters listed here. Let's do this step by step. First, if you want to remove some samples already in your report, just click here. Maybe I don't like the M5 samples. This is out of group and have the this don't have a good QC result, and I want to remove it. So unclick here. You can rename the sample's name. Maybe I want to change the M4 samples into the T4, and the T4 name will displayed in the final report instead of the M4. And if you want to filter out some genes or some abundance putting flag, you can use these parameters. And if you want to update the treatise and the environmental settings, you can set it here. And group settings. If you want to change the group settings or you want to, maybe you forget some groups or you want to add another group settings, you can do this here. Maybe I want to add a new group such as group 1 versus group 2. The group 1 containing the samples of M1, M2, M3, and group 2 containing the samples of M6, M7, and M8. Add this group, and all this group will listed in your new report. After you change the parameters, and you can submit analysis. And you can find the report updated in this part, new report. This is how to update the final report, change the parameters already set in the analysis. On the next part, I want to show you about the analysis module. I will use alpha diversity as an example. If someone clicked alpha diversity, we will have an introductory page of all this analysis meaning and the result you will obtain. We will use here, I want to use alpha diversity index. For the advanced analysis, they have the same way of submission and check. First, you can choose the analysis data or the parameters you will use. For this analysis, alpha diversity index, first step, you will choose the sample you will use in analysis. The next part is the parameters, such as the classifications. You can use UTO, order families, and the diversity index we will use choice, ACEs, and the group different tests, student tests, or the one-way ANOVA. After you change all these analysis parameters, and you can just submit the advanced analysis in BMKCloud. Click Submit. This is because my network fluctuation, but the function you can use this on your own. After you submit analysis, you can monitor the task process in here, process record. Click here. All this analysis will listed here. This is the analysis I just prepared, and you can see the statement of this analysis is finished, which means I can change this, I can download or check the result of this. If the analysis we just submit, the label here will be running. After you got the finished analysis, you can click the name and the new window will be open. And turn to the analysis report, you just submit. Okay, just I talked about how to update the report and how to do the advanced analysis in BMKCloud. Let's back to my slide. There are some advanced let's summarize here. There are some characteristics of BMKCloud in advanced analysis. The first one is freedom. You have no limitation to using advanced analysis tools. You can use it anytime and anywhere you want. And the next characteristic is customization. Users could adjust color and font to meet their specific need. You can change all this by your own. And the third part is automation. BMKCloud can automatically finish the analysis and monitor the analysis. If the analysis and all the task or the process is finished, you can check it in BMKCloud. You don't have to change the parameters or check the or upload the matrix or table the analysis need. BMKCloud will do that all this part automatically. Let's summarize of the whole analysis. This is the summary of the whole presentation. The first part I introduced about what's BMKCloud and how BMKCloud provides service in various ways. The next part we talked about task management in BMKCloud, such as how to submit, how to monitor, and how to update and how to download. The third part we went through about main results in BMKCloud using an Amplicon analysis report as an example. We went through all this part of the main results. And the last part, we talked about advanced analysis in BMKCloud, how to customize final report in BMKCloud. That's all my presentations here. Let's give the host back to Lydia.
Okay, thanks for Jonathan's presentation. He explains the topic in great detail. I believe we all have a good understanding of Amplicon in BMKCloud introduction. Due to time constraints, we apologize for not being able to answer questions. But if you have questions, you can send it to us and we will answer them by email. So we will conclude today's session. I want to express our gratitude for your participation. Look out for the follow-up email with a webinar summary. So bye, have a nice day.
Bye, thank you.